In this series of videos, we're going to take a look at the origins and diversity of members of the animal kingdom. And if you look at this figure here, you can see one of the hypotheses for how animals formed is that they are based on a ancestral proteus that was out there, more than likely one of the protozoans, because they do have these animal-like characteristics. And these cells formed a colony. And within that colony of cells that we can see depicted in this figure right here, the cells started to become specialized. They developed a variety of mutations, and that gave those cells really some different functions, and that allowed them to start doing different kinds of things that really resulted in these cells starting to become dependent on each other. Some of these cells in this little figure here you can see are used to move them around. They retain their flagella, whereas these little cells over here in this example are the food synthesizing ones. So this would be an example for the algae and going on to become plants. And the similar kind of process is hypothesized to have worked for animals that are out there, that an ancestor was able to form a colony, and then those colonies became more and more specialized, and they formed tissues. And if we take a look at a phylogenetic tree of the animal kingdom, what we're looking at here is basically a group of organisms that are characterized by being multicellular, eukaryotic. They are presumed to have evolved in the ocean probably about 600 million years ago. And when we look out there today, there are more than 1 million identified species that are still living today. Some other characteristics of the animal kingdom is they are heterotrophs. So if you remember that idea, they are getting their carbon by ingesting living animals or dead organic material that is out there. And we saw from that previous figure that these animals have developed the ability to form specialized tissues, and the cells are going to differentiate to perform really unique kinds of functions inside of these tissues. If you think about the animals that you see out there, one of the very distinct differences from things like plants and fungi that are out there is that animals are motile during their life. And the other big difference that we see is very few animals can actually reproduce asexually. When we do see that, they are usually stationary aquatic animals. So think things in the ocean that are sort of attached. And they can reproduce asexually by basically budding or breaking off or fragmenting. And then that parent is going to still continue to grow, presumably, and then the fragment that came off of them can land somewhere and grow into a new individual. There are some interesting cases when you look at some insects that are out there that can actually use what's known as parthenogenesis, and that's where an unfertilized egg can actually develop. And if you are looking at bees, the drones that are out there, what are considered the male bees, they actually are from unfertilized eggs, whereas the females are from fertilized eggs. When we look at that process of reproduction in animals that are out there, they can reproduce sexually. And so we want to take a look at this process here. And if we look at this process, again, this process of sexual reproduction is going to involve two haploid cells that are going to meet, and then you get fertilization of your egg by your sperm, and the two nuclei are going to fuse together to give you your diploid zygote, that is your fertilized egg. And this zygote right here is going to be able to divide by mitosis, and it's going to go through a series of divisions. So one cell becomes 2, 2 becomes 4, 4 becomes 8, and you can see this little ball of cells that's forming in the 8-cell stage. And what's really kind of interesting here is this ball of cells is actually going to form a fluid-filled ball of cells that we can see depicted over here in a cross-section where part of this has been chopped away, and this is known as a blastula, and it's usually around 100 cells, and in humans this is when you hit about 4 to 5 days post-fertilization. And this ball of cells here in the blastula form is full of what are known as embryonic stem cells, and these are cells where their specialization has not occurred, and effectively these cells can become anything in your body. And there is a big hunt in science to find these embryonic stem cells because they wanted to be able to expose them to various hormones and then start to get them to develop into things like organs and that. And a few years ago, a guy over in Stanford was actually able to take adult cells and introduce a couple of genes, and later on they were able to introduce a variety of proteins, and they could cause those adult cells to actually regress back to this blastula kind of stage. And so they made what are known as totipotent embryonic stem cells. Or induced pluripotent ones. So it's kind of an interesting thing that we can actually reprogram our own cells to cause them to go back to this stage where we were just a little ball of cells. Well, if you look at this process, when you take that blastula that is out there, something interesting happens when we look at animals. They are going to start to go through this process known as gastrulation, and the cells here will actually start to migrate towards the interior of that blastula. And this is showing it in early gastrulation, and then later on during this process of gastrulation, you can see there's this invagination that took place inside of that ball. And this is a really important feature when we look at the animals that are out there, because these cells now are starting to take on their role. They are starting to specialize just by coming in and forming these different layers right here. And if you look at this, the outer layer of cells that forms during gastrulation is known as the ectoderm, and this is the outer cell layer that is there. And this is going to form things like your skin. Also, interestingly, it's going to form neurons in your brain. And then if we look at this, we see there's this sort of middle layer right here that's known as the mesoderm, and this is going to form the linings in animal bodies, also things like muscles and blood cells that are out there. And then the fold that forms right here, this is known as the endoderm. Endo is inside and derm is skin there, so this literally means inner skin. And this is going to form things like your digestive system, your respiratory system, and a variety of different glands inside of your body. So this process of embryogenesis in animals is actually going through this set of graded steps, really, where we're going to start to form these different tissue layers really early on during embryogenesis, after about a week when we look at this process in humans. And if we look at this process over here, what we can see is this animal is going to take these different tissues right here and start to form these specialized tissues inside of it. And here we're looking at a larva, and this came from a figure where you were actually looking at some sea stars that are out there. And that larva can go through the process of what's known as metamorphosis. And you think about this thing does not resemble a sea star out there. But that developmental stage was set up during this process of gastrulation, and then these cells have gone on and their fate has been determined. And we learned about this because people actually take these gastrula here, and you can go in with a laser and kill one specific cell within this. So you can actually map the fate of any cell inside of this process when they go from the blastula to this process of forming the gastrula right there. So they laser ablate a cell, and then they look and see what effect does that have on the larva and potentially on the adult if it can survive that long. And so we've been able to map out the fate of all of these cells inside of this kind of ball of cells here. So it's really kind of an interesting thing that developmental biologists have taken a look at here. All right, so let's change gears a little bit and take a look at some of the advances that have occurred when we look at this phylogenetic tree of animals that are out there. And you can see, remember, when you're looking at a phylogenetic tree, each of these is a branch where you see these two groups of organisms have diversified from each other, and they've done something different, and that feature is retained in their modern descendants. So all of these animals that are out here are grouped together because of similarities in what they look like, fossil evidence, and also nowadays this idea of genetic evidence for comparison. But if we trace back through their evolutionary history, we can see that they came from ancestors that had certain kind of characteristics that we still see retained in a lot of their modern descendants here. And so if we look at the most basic type of animal that is out there, it's way down here on the bottom of this phylogenetic tree. This is where we see those colonial forms. And those colonial forms went through this branch point where you start to see this specialized tissue developing here. And if we look at our colonial form that's out there, well, this is really a simple kind of animal that people just don't think about. And what we're looking at is sponges, and this is in the phylum Porifera. So if you remember our taxonomic categories, we start out with our domain Eukarya, we're in our kingdom Animalia, and now we're down here at this third level, the phylum Porifera. And when you look at the sponges that are out there, they really are just a group of cells that have sort of come together and are doing some very, very simple functions, but we can't peel them apart like a tissue is. And you can really think of a tissue like a tissue paper you use for blowing your nose, that you can peel those things apart and you get a defined layer of cells or tissue right there. And sponges, we cannot do that process. Effectively, what they are is this sort of skeletal arrangement here, and then on that skeleton of all these fibers, you're going to find two different types of cells that came together in that colonial form. One of them is known as the choanocytes, and these guys are basically the cells that have this flagella right here, and they're going to flap their flagella, and that coordinated beating of those flagella is going to draw water into the sponge, and then it's going to push it right out through the top right here. And as that water is traveling through this sponge, the amoebocytes and some of the choanocytes are going to be able to collect some of the food that is passing through them. So think microscopic bacteria and other kinds of cells that can pass through this organism are going to be collected, and there is your heterotrophs. They're going to be able to eat these other kinds of cells that are out there. So really a very, very simple kind of animal that is presumably an ancestor form that we can still find today with their descendants. These sponges went through a couple of different changes here. We start out with this colonial form, and then we see this development of tissues going on. And so there is that branch point. And then all of these organisms over here are the ones that do have those true tissues. And one of the interesting things is now that you've developed true tissues, you're going to start to see some differences develop. So once you have those tissues, now we get another branch point in the development of our animals that are out there. And this is where biologists start to look at what's known as body symmetry. And when we look at this process here, this goes back to our idea of those germ layers that formed in the embryos that are out there. This is going to give us different layers. And so if we look at this figure, what we see is two different versions of this process right here. In these animals, they're known as diploblasts, and they're going to basically form those two embryonic germ layers. You're going to have your endoderm and your ectoderm. And this group of animals that are out there tend to form what's known as radial symmetry. And if you look at something that has radial symmetry, you can cut this and you get mirror images on either side of that plane. You can cut them in another plane and you get mirror images. So when we look at this, the symmetry is going to radiate out from the center, and you can cut them in multiple locations. So this would be things like jellyfish that are out there, or sea anemones. The triploblasts over here are a little bit different because they actually do form that mesoderm or middle layer right there. And animals that come from this origin here that are triploblasts have what's known as bilateral symmetry. We can cut them in one plane, and that's going to give us equivalent mirror images for the left and the right. And so again, this third layer is going to start to give us things like a head and a tail that is developing that idea of bilateral symmetry. And so if we look at some of our simple kinds of animals that are out there that have this radial symmetry, the one group or phylum again is what's known as the cnidarians. And these guys are the diploblasts that have those two tissue layers, and they have that radial symmetry. And if you look at these two forms of them, they're very similar. They're just inverted from each other. The polyp form is going to attach on the bottom, and then its mouth butt is up here on the top, and then you form this gastrovascular cavity right here. These guys are what's known as the medusa form, and this is what we're familiar with from jellyfish. And they're basically just inverted, so they're floating around in the water. And when you look at them, those two layers of tissue are going to form this outer skin and then this inner kind of stomach organ right here where they can digest their food that they shove into this one single hole using their tentacles. And on the ends of their tentacles, all members of this cnidarian phylum have what are known as cnidocytes, which are stinging cells that can have toxins. So they can actually use that to stun their prey as they shove it into their mouths.